Hello everybody, this is the grade 9 biology review for the trial 1 exam, semester 1 at the International School of Innovation. Mainly, the cover chapters are digestion and some types of nutrition. So, chapter 1, activity 1, it's about our food. So, what is the food that we eat? What does it contain? What happens to the food when we eat in our body? What is the final fate of the food? To start with, we have to talk a little bit about the definition of digestion. Digestion, simple definition, is the transformation of complex food molecules into the simplest one. This is the complex food molecules. You are digested into simplest one, used to be used by the body cells. Mainly these are called nutrients. So, which system is responsible for the digestion of food? As we know, it's the digestive system for sure. So, digestive system mainly is divided into two main branches. The first branch is called digestive tube, and the second is called digestive gland. What is the digestive tube? It's where the organs through which food can pass through, while digestive glands is organs through which food doesn't pass through. Organs of the digestive tube and the digestive glands. Digestive tube, we have the mouth, then we have the esophagus, stomach, small intestine, large intestine, and finally we have the anus. While digestive glands, we have salivary gland, gastric gland, the stomach, pancreas, liver, and we have intestinal cells or intestinal glands, sorry. So let's move here to the function of the digestive gland. Digestive glands, they are known to secrete digestive juices into their site of action. What is this? Digestive gland. First, salivary glands. What do they secrete? They secrete saliva, where at the level of the mouth, where it is neutral, meaning that pH is equal to 7. Gastric glands, they secrete gastric juice at the level of the stomach, where it's acidic, meaning that pH is less than 7. Pancreas secretes pancreatic juice at the level of the small intestine. Liver secretes bile at the level of the small intestine. Intestinal glands, they secrete intestinal juice also at the level of the small intestine. Those three, they are the level of the small intestine. The medium is basic, meaning that pH is greater than 7. So, let's move now to what the meaning of neutral. Neutral medium is meaning the mouth. Salivary gland, they secrete the saliva at mouth. Mouth is neutral, where pH is equal to 7. Acidic, pH is less than 7. Basic, pH is greater than 7. Foods, they are divided into organic and inorganic. Example about organic: carbohydrates, proteins, lipids, and vitamins, while inorganic, water and mineral salts. Let's move to this table. Category of organic food. We have mainly, while talking grade 9, about carbohydrates, about proteins, and about lipids. I'm going to talk about the complex form, the intermediate, and the simplest form in the coming slides. So now we can go deeply into the definition of digestion. So digestion is the set of chemical and mechanical transformation of complex food molecules into simpler nutrients. These nutrients, they are going to be used by the body cells to produce energy needed for the activities of the cell, which is known by the cellular activity. Category of organic food. Pay attention to this. You have to study this table. You have to know every category of organic food. Which one is the complex, which one is the intermediate, and which one is the simplest form of food. So carbohydrates, they are natural source of sugars, fat sources of energy. They contain carbon, hydrogen, and oxygen. The complex form of carbohydrates is known to be the polysaccharides. Polysaccharides is the complex chain of simple sugar. We have starch as an example. We have cellulose, which is an example of carbohydrates in plants, and we have glycogen in animals. Intermediate is known to be di. Di meaning that two. So it's contained two simple sugars. They are bounded together. First example is maltose. it's glucose plus glucose. Second, we have lactose, it's glucose plus galactose, mainly in milk. Sucrose is glucose plus fructose in fruits. Last one, simplest form or the nutrient form, it's the monosaccharide. Mono, it's only one. So it's made up of one unit only. Example, glucose, fructose, galactose, all of them, they are reducing sugar. Moving to proteins. Proteins, they are called plastic food. Proteins, they are complex chain of more than 52 amino acids. Polypeptides, this is the intermediate, it's less than 52 amino acid, while the simplest form is the simplest form, which is made up of only one unit. Lipids, they are very energetic. The same quantity of lipids have a double the amount of energy that's supplied by carbohydrates. Complex form is lipids. Intermediate, there is no intermediate, and the simplest form is fatty acid plus glycerol. How to identify the presence of certain categories of food. First, we have food test. What is the aim and step of the test? What is the positive result and what is the negative result? And finally, analysis of the positive and the indication. Let's start one with water test. Water test, from its name, it's aimed to identify the presence of water. Steps of the test, you have to heat the food in the test tube. We have to heat it. Positive result is the presence of water droplets on the wall of the test tube. This is a positive result. While if it's a negative, no water droplets are formed. So more water droplets at the walls of the test tube. Analysis. After heating the food, water droplets are formed on the walls of the test tube. What does this indicate? This indicates the presence of water in the food. Test number 2 is salt test. Salt test, it aims to identify the presence of sodium chloride. We have to add distilled water to the food in a test tube and we have to shake it well. Add silver nitrate AgNO3 to the test tube. White precipitate is going to appear, which is called silver chloride crystals. Negative result, no white precipitate will appear. What does this analysis? Analysis after adding distilled water and silver nitrate solution to the food, white precipitate is going to appear. What does this indicate? This indicates the presence of salt in the food. Number 3, pay attention to this one, Fehling test. Fehling test is mainly for the reducing sugar, glucose, fructose, galactose, maltose, lactose, and so on. So we have to add equal numbers of Fehling, NaOH, and Fehling B, CuSO4. Heat the contents of the test tube. Positive result, brick red precipitate appears. Negative, solution is going to remain blue in color. Analysis, after adding equal drops of Fehling A, which is NaOH, and Fehling B, CuSO4, and applying them to Fehling test, brick red precipitate is going to appear. What does this indicate? This indicates the presence of reducing sugar in the food. So brick red precipitate is an indication of the presence of reducing sugar. Iodine test, which is test number 4. Iodine test is aimed to the presence of starch. What do you have to do? We have to add equal drops or few drops, of iodine solution. If dark blue color appears, this is a positive result. If no dark blue color, mean that orange, yellow, brown, or yellowish color mainly, so it's a negative result. After adding iodine to the food, dark blue color appears. What does this indicate? This indicates the presence of starch in the food. Pyrite test. Pyrite test is done for the presence of proteins and or polypeptides. How can we do this test? We can do it by adding equal numbers of drops of NaOH, which is colorless, and CuSO4, which is blue in color. What is the positive result? Violet color is going to appear. Negative result, color is going to remain blue in color. What is the analysis? After adding equal drops of NaOH and CuSO4, or after applying pyroTest, violet color. So the key here is the violet. So proteins with violet test, they're going to give violet color. What does this indicate? This indicates the presence of protein and or polypeptides. And pay attention to this one, and or polypeptides. Moving to coagulation test. It aims to the presence of proteins. Heat food till boiling. If it's coagulate, so it's a positive result. If it doesn't, so it's a negative result. After heating till boiling, the food is going to coagulate. This indicates the presence of proteins. Translucent spot test. It aims to test the presence of lipids. What we have to do is to rub a piece of food against a paper. Observe it under the sunlight. If a translucent spot appears, this is a positive result indicating the presence of lipids. No translucent, so no lipids. After rubbing the food against the paper and observing it under sunlight, a translucent spot is formed. What does this indicate? This indicates the presence of lipids in food. Sodium test. Sodium test indicates the presence of lipids. We have to add sodium test solution. A pink color is going to appear. It's a positive result indicating the presence of lipids. If no pink color, so no lipids. After applying sodium test, pink color appears. What does this indicate? This indicates the presence of lipids in food. So these tests, you have to study them. You have to know the positive result and the negative result. What does each test aims for? Aim for? It aims for the presence of lipids. It aims to test the presence of carbohydrates. It aims for test the presence of reducing sugar. You have to know this and you have to study them very well. Before moving to activity 2, you have to keep in mind that digestive juices, all of them, they contain digestive enzymes, except for the bile. What are enzymes? Enzymes are the biological scissors that digest or break down or cut or transform or hydrolyze. All of them, they lead us to the word digest. Digest food. They are biological catalysts that speed up the chemical reaction without being consumed and remains unchanged at the end of the reaction. Moving to chemical transformation of food, which is activity 2. Chapter 1, activity 3. Enzymes are the agents of digestion. So, you have to keep in mind here, optimum temperature for the working of enzyme is 37 degrees Celsius. Enzymatic activity is best at this one. That's why it's called optimum temperature. Inactive temperature is 0 degrees Celsius. Since enzymes, they are inactive at this temperature, activity is 0%. 60 degrees is the denaturation or the destruction temperature. Since enzymes are destroyed under high temperature, so they can no more act. The activity is going to be 0, even though we return them to the normal temperature, which is 37 degrees Celsius. You have to study here the table of digestion in organs. Every enzyme, on which substrate it acts, at which pH, at which temperature. If an enzyme is inactive at cold temperature, but is replaced at normal temperature, which is 37 degrees Celsius in this case, in human body, so it becomes functional again. While if an enzyme is placed at very high temperature, then replaced at normal temperature, let's say very high, about 70 degrees Celsius for example, and then replaced at normal, it won't function. Why? Because high temperature will denature the enzyme. So the enzyme is denatured, it can't function again. We have to work about the mode of action of some of the enzymes. Mode of action by simplification of salivary release. Pay attention to this. You have to give legend. You have to give the legend. This is all. This chain is starch. Every one of these, every two, let's say it's maltose. Every one of these is glucose. You have to give the legend. This symbol is for the enzyme salivary amylase. This one is for the molecular simplification of proteins by the enzyme pepsin. So every one of these, every triangle is amino acid. This enzyme is pepsin. and every 3 they go for polypeptides, or every 4, every 5, and so on. So you have to give legends, you have to draw it using a pencil, you have to give it a title, and you have to write the legend in a clear way, and you have to use it. Third, molecular simplification of peptides by trypsin, the same idea. You have to give legend, you have to give the symbol for the enzyme, let's say, and it's not should be like this. Can be arrow, normal arrow, 2 arrows, can be, for example, like this. You can draw whenever you want. You can draw it simply like this. This is an arrow. You can use whatever you want. I use different shapes and different. This is going also for the simplification of lipids by the enzyme lipase. Also the same idea. The simplification of maltose by maltase. This is the easiest one. The enzyme maltase, and this is glucose. So maltose is 2 glucose together connected. Moving to chapter 1, activity 4, from food to nutrients. If our digestive system is capable of digesting the food we eat, no matter what, and transform it into simple nutrients, then why do they insist we have to chew our food well before swallowing? It's easier for the digestive system to digest small pieces than digesting large pieces. This is the big answer here. So it's easier to digest small pieces than digesting large pieces. So digestion can be divided into mechanical and chemical. Mechanical involves digestion of food into smaller pieces. Chemical involves the action of enzymes. So mechanical digestion is the digestion or the breaking down of food into smaller pieces. This will keep food moving through the long digestive tube. Organs where mechanical digestion involved. Mouth, we have something called chewing of food. And the level of the stomach, we have called churning. Small intestine, we have something called peristaltic movement. Chemical digestion. Chemical digestion mainly involves the digestion of food by the help of the digestive juices. They break down food into simple nutrients. It occurs in the mouth, in the stomach, and the level of the small intestine. How are these 2 types of digestion needed? Mechanical digestion, you have to keep it in mind that mechanical digestion facilitates, this is the big conclusion here. Mechanical digestion facilitates, so digestion of food, or the cutting of food, or the breaking of food into smaller pieces is going to facilitate the action of the digestive juices and the enzymes present in these juices to digest food faster. The routes of nutrients. Routes of nutrients, activity 5. So we have to study the characteristics that favor intestinal absorption. We have thin wall of intestinal cell, large surface for exchange, highly vascularized, meaning that contain rich in blood vessels and lymphatic vessels, and finally its moisture. What are the different routes of absorption? We have blood, we have lymph. Blood, it absorbs all the nutrients except for fatty acid and glycerol, while the level of the lymph, it absorbs only fatty acid, or mainly fatty acid plus glycerol. Pay attention to these notes. Pay attention to the notes that I covered in this presentation.